Okay, and I'm going to have to be a little bit, let's see if I can actually, oh, it doesn't show it on there. Shoot. Up this way. I left my pointer on the cart that's over selling baked goods for the Tri Beta Halloween sale. I had it on the cart that I brought to the Bat Talk. So thanks for coming to that if you did. Your points are in the mail, sort of. I have to make another column on Blackboard and then I'll put them up. So I'm going to do it this way rather than, there goes another bug. I'm going to do it this way rather than full screen because I don't have a pointer on me. So the confusion, besides learning the proteins that do all the jobs, which is a thing in and of itself, a lot of the confusion I find in chapter 10 comes from this. So this is a replication bubble for linear DNA, but really whether there's linear DNA or whether it's circular DNA using theta replication, it's the same thing. So if it's linear DNA, you would just have DNA off all the way over here and all the way over there to the end of the molecule. You would be coming, each of these forks would be coming towards another replication fork coming in the opposite direction. If it was theta replication, it would just be like this, a circle going around on the bottom that is closed there. But in either case, when I ask students about putting leading strands and lagging strands onto a replication fork like this, there's confusion. The way that you do this is you have to look at the template strand. And you have to remember what I've tried to hammer into your brains about how DNA synthesis works. So you have to remember two things. First, that the DNA double helix is an anti-parallel arrangement. So what does anti-parallel mean? Right. Opposite, the two strands are in opposite orientations. And what do we mean by orientations of the strands? We mean which end is the 5' end and which end is the 3' end of each strand. In this one, the top template strand is going 5' to 3' left to right. The bottom template strand is going 5' to 3' right to left. So then, once you get past that, the next thing is figuring out what the leading strand and the lagging strand is. And so here you have to remember that each bubble has two forks. If I were only to give you a replication fork, one fork, then it would either be the left half or the right half of this diagram. But if you see something that's the whole thing and the origin of replication is marked, you know that when the whole gray DNA strand was together, right here is where replication started. So one fork, the one on the right, starts from here and was moving to the right. The other one starts from here and was moving to the left. That's important for figuring out what's going on over here. So let's figure out, let's consider for a minute the replication fork that's moving from left to right here. So if those strands weren't there, you would just have to put, you would have to figure out which way things are going. So let's look at this, and I'm going to then go to the iPad and just draw a blank one, and we're going to see if we can try to put all this stuff in there and then also add in all of the other proteins and primers and other whiz-bang stuff that we talked about. My iPad went to sleep. All right, so let me shift this. Okay, so let's draw that giant replication bubble again. And this time there's nothing on it. So over here is DNA that's still base paired. Over here is DNA that's still base paired. All right, so here are the origins of replication. And here's the polarity of each of the strands. All right, so first, let's consider the right-hand replication fork of these two, and we're going to go through it together and fill stuff out. So let's consider this template strand right here to start with. So we're going to have to pretend that we're back to the beginning when this replication fork wasn't open quite this wide, right? When the replication fork first pops open, that's done by the initiation proteins. Remember that? So after the initiation proteins, what is the first protein that comes in and helps to separate the strands? Yes, DNA helicase. So I'm going to tell I'll tell you the joke and hope so you won't forget. So you're in the bar, right? And you meet somebody, and you know, hey, oh, what a nice person, and you know, and so you get to talking, and you know, because you really have nothing else as good pickup lines, you ask them, what if you could be any DNA if you could be any biological molecule, what would you be? And you listen politely while they say whatever it is and nod, and then you wait for them to ask you the same thing. What? Hey, but it's the biology nerd bar. So, it's a biology nerd bar, and they're serving non-alcoholic beverages to all of you who are under 21. Anyway, so, and then you wait for them to ask you the same thing, and you say, I want to be a DNA helicase, and they ask, and they say, really, why? And you say, so I can unzip your genes. Thank you very much. And with that, nobody ever forgets. Nobody ever forgets what helicase does after. And that's the only, I have others, but they're not appropriate for they're not appropriate for a class setting. Anyway, so there. The helicase comes in, and remember, it's the hexameric molecule, so we'll draw that up here. It's gonna come right around here, you know, around one of the two strands, and it burns ATP to move down the DNA strand, but it's double stranded at the time, so we'll draw it right now up here. And it is using ATP to push against the part of the DNA that is still double stranded and break those hydrogen bonds that's holding the strands together. Sorry for the bad writing. If I'm left-handed, if the palm of my hand touches the board, it won't let me write somewhere else. All right, so there's DNA helicase there, and I'll just draw it over here too. All right, after DNA helicase goes through, then it leaves the double-stranded DNA as two single strands of DNA. Before anything else happens, what has to come in and protect those? The single-strand DNA binding proteins, right? Let's make those brown. So, you know, right? Not great writing because it's over on the right. So single-strand DNA binding proteins come in. And the single-strand DNA binding proteins are always going to be there because there's always going to be DNA opening up. Now, as soon as the other proteins that we're going to talk about come through and new DNA is synthesized on that template DNA, which is this right here, you know, template DNA, as soon as new DNA is synthesized there, that act displaces the single-strand DNA binding proteins because they're no longer required. But as they're getting kicked off one spot, they're going to be added new spots down the line, right? Okay, so as we first open up the DNA molecule on both strands here, but we're going to look down here to start with. How do you get the DNA synthesis started right here at the origin of replication? Can DNA polymerase do that? Just come in and start putting DNA nucleotides next to the ones that are already there? Why not? Because it can only work with what? DNA polymerase. So you're talking about primase. We'll get to that in a second. DNA polymerase needs the primer made by primase, but before we get to that, why does DNA polymerase need that? Why can't DNA polymerase, you know, what does it need to work with that's not there right now? It needs a free 3' hydroxyl group at what's called, do you remember what that was called? Where at the primer template junction, right? So what is a primer template junction? Talked about it last time. It is the spot where if this is the template DNA, what does primase do? It comes in and puts an RNA primer where you're going to get DNA synthesis started. It's going to be anti-parallel. The black here is the template DNA. The green is the primer that primase is laying down, and its RNA sequence. And here at the 3' end is a hydroxyl group. So the primer template junction is this right here, where the double-stranded ends and the single-stranded begins. So while primase was able to come in and lay down this RNA primer where there was nothing there before, DNA polymerase needs a primer template junction with a 3' hydroxyl in order to be able to do DNA synthesis. So what is the significance of the 3' hydroxyl here? How does the chemistry of adding a nucleotide, a deoxynucleotide triphosphate work? So let's pretend, I'll go back to the other thing in a second, but while I got this here, we'll pretend that the primase just laid down this RNA primer, it went away, and DNA polymerase enzyme, DNA polymerase 3, is going to come in and add the first nucleotide. So what's the significance of the 3' hydroxyl group there? So it can, right, it can bind, but what does the 3' hydroxyl, so you're going to bring in a nucleotide, right? A deoxynucleotide triphosphate. What end of the nucleotide triphosphate is going to join over here? Right. Remember that? So here's your that's your base, and then here's the 3 phosphates up at the 5' end, and there's the 3'. This 3' hydroxyl attacks the alpha phosphate, which is the one that's closest to the sugar. Yes? Oh, sorry. It was said it there, and so I don't know why. Maybe I went to the new window or something. Yeah, thanks for reminding me. It stopped. Yes, so the 3' hydroxyl has to attack the alpha phosphate, which then causes. Can everybody can see this color of yellow, okay, right? All right. These 2 phosphates, and then they go off over here. And eventually they get split up, and that's the energy that drives the reaction. And because the 2 phosphates are gone there, now what you've got here is this is wrong, but you know, the base would be down. The base would be down there interacting with the other base. So the bond, what I'm trying to show is the hydroxyl becomes loses its hydrogen, and the former OH becomes joined to the alpha phosphate there to make that phosphodiester bond that holds the new nucleotide in. So that's why you need the free 3' hydroxyl at the primer template junction to get that going. All right, so we're going to add those in there. Let's go back here. So the first thing that happens is that what enzyme do we say comes in and adds the RNA primer? Single-stranded DNA binding proteins are binding to the DNA to keep it separate and keep it from getting attacked. They're going to be gone away once what we're talking about happens. So you need the primer here, this. What enzyme? Because DNA polymerase 3 can't add a primer to DNA that doesn't have a primer template junction. So the RNA primer has to be added by what? Yeah, so primase. That's the enzyme back here. So all right, so primase work comes in, and then DNA polymerase comes behind it, right? DNA polymerase 3, is going to come behind it, and let's see, what color should we make the DNA? The new strand of DNA is going to be purple. So green is going to be RNA and purple is going to be the new DNA strand. Comes in like this, but as it's coming through, these single strand binding proteins are displaced. And of course there will be more of those down here as soon as helicase opens up more sequence. So right, so that's the 3' end of that newly synthesized strand. But you can see that this strand is elongating in this direction, right? The same direction that the fork is unwinding. So which strand is this going to be called? This is going to be the leading strand because it's continuously synthesized. Okay, what was your question? Well, okay, so we'll, let's see, what color can we make? We'll make the gyrase yellow so we can use it somewhere else too. Gyrase will be somewhere probably off the screen, but we'll just draw it here and here. So what is, as long as you're mentioning it, what's the job of DNA gyrase? Prevents excess torque. When you open up the DNA strand, it creates supercoils. Gyrase relieves them. So as a strand is, as a leading and lagging strand is moving, this the replication fork are moving. This replication fork is going right. This one is going left. The first protein to act way out in front of where the replication fork is actually splitting the double-stranded DNA into single-stranded is DNA gyrase. So that's the first thing that's going to act on a certain area of DNA before the DNA strands even get opened up. The reason for that is because if it doesn't, then you'll get so much tangling that those strands, the two DNA strands, will never be able to open up and they won't be able to be used as template. All right, so that's the leading strand. You notice that there's only one RNA primer on it, right? Why does DNA primase only work one time on the leading strand for a given replication fork? Yeah, because the DNA polymerization is continuous. You always have a free 3' end, so DNA polymerase 3 can always do its job, and you're good. All right? So let's move up here to the top now. Let's work on this area. So when the origin of replication first opens up, nothing can happen around right here until about, let's say, like this much DNA sequence is exposed. Now this isn't to scale. It's probably 200, 300, 400 bases. I don't know exactly how long in each species Okazaki fragments are, but that's in the ballpark. So you have to have that much DNA actually get split into single strands before you can even start here. That's why this strand is going to have the other name, which is what? The lagging strand. And you can remember the names leading and lagging because the lagging strand, synthesis on the lagging strand lags behind synthesis on the leading strand. And the reason is because you have to wait until enough DNA gets opened up to actually synthesize on that strand. Right? So so once you get to right here, then primase is going to act there and put a primer. like that, right? And in this case, the 3' end is to the left. So why is it to the left where when the bottom strand primer is pointing the 3' end to the right? This primer is pointing to the left. This one is pointing to the right. Right, because the orientation of this template strand is opposite the orientation of this template strand because they were anti-parallel before they separated from one another into single strands. So this one on the template strand on top has 5' at the left and 3' at the right, so that DNA synthesis, which always takes place in which direction? 5' to 3' with respect to the DNA being made. So you have to go here, and then DNA polymerase III would go over and stop there. The reason why it stops there is because on this strand, because this strand is the other leading strand, you've already got a primer there and it runs into it. We'll get back to that in a minute. All right, so you've got this, and then where's the next? So on this upper strand here, where's the next primer going to be? Yeah, right over toward the right there. So like, let's just put it like right here. And then the second Okazaki fragment is going to be synthesized by DNA polymerase III like this, and it goes down and it stops right there. The reason why it stops is because it runs into the primer from the previous Okazaki fragment. So as you go through here, primase, which makes RNA primers, is going to work many times on the lagging strand, but only once on the leading strand because, well, you said synthesis on the lagging strand on the leading strand is continuous. Synthesis on the lagging strand is discontinuous because you have to keep waiting for more DNA template to open up so you can do that. Good? All right. Let's fill in the other side. I already filled in here. So now DNA synthesis is going to take place in a 5' to 3' direction, and it's just going to go like that, keep going that way. This is also going to be the leading strand. So you could just sort of, if you have to try to memorize something, which I really don't recommend, you can remember that the leading strands are like catty-cornered from each other or diagonal from each other. They're not right across from each other. I think the main thing to remember is that the leading strands and lagging strands are not both on the same side here and then the other one's on the same side there. And the way you figure it out is look at the template strand. Remember which direction DNA is always synthesized and put it in there. Yes? Well, I wouldn't say that it would, but if I, well, I mean, it's important. I'm emphasizing it as an important point and taking up more class time to review it, which means that I ask questions about almost everything that I do, So this is going to be the other leading strand here. That's the lagging strand. So that means over here at the bottom, this guy at the bottom is going to be the opposite of that. So the primers here are going to be like this, going that way, here, and like this, going that way. All right, so we've already talked about primase and show that primase works to make RNA primers. Where does DNA polymerase III work? That's DNA polymerase I. DNA polymerase III, what's its job? It's just the main DNA, the workhorse we call it, DNA polymerase, that elongates the strands. So DNA polymerase III is going to be, we'll use yellow for that. Well, let's pick another color. Let's use orange for that. So DNA polymerase III is going to be wherever you have a 3' end of newly synthesized DNA, right? Make sense? Right? So now let's go and talk about what we were just bringing up. So DNA polymerase I. What is the job of DNA polymerase I doing? Right, so it has two functions. What are the two functions? It removes the RNA, but what's the enzymatic name for the function? Does anybody remember? It is an exonuclease, right? So an exonuclease, it's got a 5' to 3' exonuclease function, so it can remove the RNA primers and make them into DNA. So let's kind of out of colors. We'll use blue for that. So here, at the beginnings of each, that's too close to the helicase, but at the beginnings of each primer, the 5' end of each primer is where DNA polymerase I is located. And DNA polymerase I has the 5' to 3' exonuclease function and also has a 5' to 3' DNA polymerase function. So DNA polymerase III and DNA polymerase I both have the 5' to 3' polymerase function. That's how you add nucleotides to the chain. But DNA polymerase I does not have the 5' to 3' exonuclease function. Okay? Then after, let me go over here. So after the DNA polymerase I comes in, it will use its 5' to 3' exonuclease function to remove these bases. But as it's removing each of these bases, it's also using its 5' to 3' DNA polymerase function. So the DNA nucleotides are being removed, or RNA nucleotides are being removed, and DNA nucleotides are being put in. So what ends up happening then is one by one, you're removing the RNA nucleotides, adding DNA nucleotides, like that. until you've got a new DNA molecule in there. But at the end, you are left with this. So there is base-to-base contact all through this area. This is the last nucleotide that was added. The base is there, and the next base is over here, but what you're missing where the green arrow is a phosphodiester bond. And so what is it called when you have one strand, the bottom one is intact, and the top one is missing one phosphodiester bond there? A nick, right? That little gap is called a nick, right? And so this requires an enzyme to come in and fix things up. So at this end you have actually it's only one phosphate because it was hooked to the RNA. So there's one phosphate there, and up here is a hydroxyl. So what you need to do is have this oxygen attack the phosphate so the hydrogen can go away and you make the phosphodiester bond. What is the name of the enzyme that does that? That's DNA ligase. So this is the very end. So as you have a replication fork going in a certain direction, let's go back here. This replication fork here is going from left to right. What is the first protein that acts on the DNA way out ahead of the replication fork? The gyrase, right? That's taken out the supercoils. Then what is the protein that's acting right where the two strands are separating? The helicase. And then after that, on the leading strand at least, you have the enzyme that is adding new bases as fast as the DNA is being unzipped or opened up there. And what is that? Well, single-stranded DNA binding proteins, right? But mostly once this is going on and opened up, I'm going out on a limb here, and I'm guessing, but I would say single-stranded binding proteins, once this is all set up and started, are going to be somewhat on the leading strand, but almost exclusively on the lagging strand. Let me get your question, and then I'll say why I think that is. Right. So you're right. So initiator protein is required. Yes, but initiator protein comes here and then makes it so that it first opens up this little mini bubble to start with. So that's the first protein that acts, but the question I was asking was when you have a replication fork already set up. So initiator proteins only function once, and they only function when each origin first fires or opens up, like going from the top of the screen to the bottom there. Once that happens, the initiator proteins go away and they're never involved again. Remember the licensing that we talked about in eukaryotes? So in prokaryotes, there's only one origin of replication. In eukaryotes, there's many, but licensing has to occur during S phase in order for than origin to open up and attract these the initiator proteins. And once that origin has opened up, like as shown here at the bottom, that's it. In S phase and G2, those licensing factor proteins can never bind again. So once the origin first opens up, then you're right. What color did we make it? Blue. Then the first thing that's going to come in here is the helicase and start opening it up a little bit more. So does that make sense? All right. So we don't cover this in that well, I'm just going to tell you, but yeah, I won't ask you questions on it. The reason why I'm saying that I'm pretty sure that single-strand binding proteins, once everything is moving, only are involved on the lagging strand is because the speed with which helicase opens up the DNA is actually directly driven by how fast the DNA polymerase can go. The helicase can actually open up the DNA faster than the polymerase can go, but you wouldn't want that because it would get way out ahead, and then the single strands, even with the binding proteins on, could get into a lot of trouble and tangle up and whatnot. So the helicase sort of is going along, and then it'll just sort of slow down. But the DNA polymerase on the leading strand is right behind it and bumps into it. And it's actually interaction with this bumping into it of the DNA polymerase active subunit or part bumping into the helicase. It's like, oh, you know, like speeds up again, right? It's like the helicase is hanging out in the left lane on the highway, and then the polymerase is coming up behind, like flashes the lights, honks the horn. He's like, okay, I'll speed up. He gets way out ahead because he can go faster, and then he slows down, and then the car's like right on his tail again. Come on, and honks and flashes the light again. That's exactly how it works. It's like everything in biology almost is a homeostasis mechanism, right? So the speed is determined by that. And for that reason, the polymerase is always right behind there. So maybe single-strand binding proteins on the leading strand interact while you're working on it like this, but just for a little bit. But on the lagging strand, you have to open up a whole Okazaki fragment's worth of template DNA before you can have another primase laid down, or primer laid down by primase. And so therefore you're always going to have an open strand. Yes, this one gets filled up, but by the time this one gets filled in, the next one is completely open. So on the lagging strand, there's always a good amount of single-stranded DNA opened up. Yes? You mean a left-handed or right-handed helix? No, because it's unwound like this. This is where it's unwound, and so the only helix. So left-handed versus right-handed helices. The only left-handed helix we talked about was zDNA, like the zigzag DNA, in certain DNA sequences where you have a purine, pyrimidine, like CG, CG, CG over and over, and then it would be like AT, AT on the other, on the other strand. And we're not even sure whether that really makes a left-handed helix in vivo. We can do it in a test tube and make a crystal and show that, but you don't have to worry about that. And the difference between the A DNA and the B DNA is just the hydration of it, and really in real life it's sort of like right in the middle. Instead of being 10 bases per turn like A, or 11 like B, it's sort of like 10 and a half. Maybe I just said that backwards. I think I did. So it's 10 bases for B and 11 for A, because the A is like shorter and more squat sequence. But in either case, when you open them up, it looks the same exactly like this. Man, maybe it's just because the doors have like cracks in them and stuff, but there is like bugs flying around the screen and bugs flying everywhere. What? No, there's like bugs up by the project. You can't sit. There's like, they keep landing on you? Okay. All right, well, I had to turn off the recording here, but so does everybody get what we're doing with this now, right? So by going through and practicing drawing everything in and seeing what everything does when it, when it works there, seeing the order in which all the proteins function as the replication fork moves along, and the one on the other side is going to move and be doing just the opposite, that will help you understand what all of the proteins do versus just trying to memorize them. And that's the point. I'm trying to get you to figure out how, or to be able to explain and understand how all this stuff works. And it's a really cool thing. Let me get this open, and now there's a fly right on the screen. And I'll go through the video with you. See, Blackboard takes forever for me too. It's done. Oh wow, this is just well, maybe I won't be showing you that video. This is that's pretty bad. So I'll try logging out and logging in again, because they've told me too that if there's 4 servers that do Blackboard, and if one of them's having a problem, you just quit the browser and try again, and oftentimes you can get the other one, because once you've got a certain one, then you're logged in. Well, all right, so I'll just leave this to try and load, and we have about 20 minutes left in class that I can answer any questions that you guys may have, so I don't go on to new material before the exam. So remember, there is some material from Chapter 8 on this, the part that occurs after the translocation. So the translocation heterozygote that does the alternate and adjacent 1 and adjacent 2 segregation at meiosis 1, that is not on this, but everything after that in Chapter 8 is. All of Chapter 10, 11, and 12 then too. All right, let's see if you guys can get sound out of this.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex.
All right, well, let me go back and I can just explain it to you then. It's easy enough. So this video is old enough that you will actually see the DNA polymerase only has 2 enzymatic heads on it, and the other one is going to, our current understanding shows that there are 3.
During DNA replication, both strands of the double...
All right, so here is a replication fork. Down here And this is on the web, this is on the Blackboard page. You can go home and listen to it with the very nice volume that actually works there. So down here you have DNA that's, here let me go back and look. So
helix act as
down here you
templates
have
for the
DNA
formation
that's
of new
being unwound.
DNA
You see the blue
molecules.
thing is going to be
Copying occurs
DNA
at a localized
helicase. It's
region called the
unwinding the
replication
DNA.
fork,
See over
which is a
here,
Y-shaped
this
structure where new
subunit
DNA
is
strands
always
are
in
synthesized
contact with
by a
the
multi-enzyme
single-stranded
complex.
DNA. It's going right into
Here, the
double-stranded
DNA to be copied
It's being
enters the
unwound here,
complex
and
from the
and
left.
getting
One new
getting
strand
rewound
is
there.
leaving at the
This is the
top of
lagging
frame,
strand. You
and
can
the other
see
new
that
strand
it's
is leaving at the
making a
bottom.
new strand there. Oh, there's
The
the lagging
first step in
strand there.
DNA
So,
replication
here's
is
the
the
lagging
separation
strand.
of the two strands by an enzyme called helicase.
Cooperate with me here.
This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated
So see,
strands are
here's the
called
lagging
3'
strand that's coming out, and
and 5',
now it's going to be worked on again. While
distinguished
new
by the
lagging strand is
direction in which their
made
component
out.
nucleotides
Meanwhile,
join
down
up.
here the leading strand just keeps working.
The 3' DNA strand, also known as
Wait
the
to
leading
stop right here
strand,
until I get
is
it going
diverted to a
resolved.
DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix.
They make a big deal about saying that
The other
the
half of the
helicase
DNA
rotates
double
faster
helix,
than a jet
known as
it's when you get into the molecular biology and when they start to do crystal structures of things, it's amazing, these little machines. What I didn't tell you about in this class is that here's, well, let me go forward a little
the
bit.
lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism.
Oh, this is showing all the three of them. So over here is the leading strand. Here is the lagging strand. One of the two heads that's actually making double-stranded DNA out of this single strand, goes all the way around here and comes back there. This part we don't talk about in this class, but this part is what connects the two heads together, and it puts these little guys on there. So what I don't tell you about is there's another little circle donut that gets put on there that helps the polymerases stay in place. And this part down here loads these little clamps or little circular pieces of DNA on there. But what you can see is now here's the one head on the lagging strand synthesizing DNA, right? And then here's more lagging strand DNA getting paid out here, and that's not going to be worked on until enough gets out and another primer gets put on there. What's not shown, because they didn't know at the time, is the third head. So this purple part is the enzyme that's actually doing the job, and then this blue thing here is called a flexible linker, and that's just holding the enzyme head to the bottom part here that puts the clamp on. See, aren't you glad that this is the stuff you don't have to know. There's always more. While this is working, the third lagging strand head would be coming over here getting ready to start the next lagging strand. So let's look again.
As it emerges from the helicase.
So see, here's one primer up here, then there's an Okazaki fragment being formed, and as soon as the polymerase gets here to the next primer, it would stop, and by that time another Okazaki fragment would be starting here and going that way until it hits this one. And then while the polymerase is working on this one, another little bit would be opened up and put out there. Does that make sense? You should go back and watch this.
The lagging strand is organized into sections called Okazaki fragments.
It's weird when you first see it.
These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation.
But this little guy
These sections are then effectively
you can just see
synthesized
the
backwards.
helicase just
When the
barreling
copying is
through that
complete,
DNA and just
the
pulling
finished
it
section
through
is
there,
released and the next loop is
it
drawn back
burning
for
a
replication.
fairly substantial amount
Intricate
of
as this mechanism appears, numerous components have been deliberately left
ATP
out
to
to
do
avoid
that.
complete confusion.
So this is right here. See this little bit from there to there? This is the leading strand coming right off, and it's immediately being worked on. The polymerase is that close to the helicase. And like I said, it will keep bumping it and then not bumping it and bumping it and then not bumping it. You guys have a question about what's what? No? Come on. So once this is done,
The
I'll go back and
exposed
watch it
strands
again
of
so you
single
can see
DNA
it's-
are covered by
it keeps
protective
going.
binding proteins, and in some systems, multiple Okazaki
The
fragments
little
may be
green
present.
clamps are being loaded on the DNA for each Okazaki fragment. It's kind of cool. All right, so let's look through like this.
During
asked them to
DNA
make sure that sound
replication,
works on
both
this,
strands of the
and
double
I thought
helix
it did
act as
previously. I'll
templates
have to
for the
ask them
formation
again.
of new DNA molecules. Copying
So you
occurs
can
at a
see this
localized
little
region
piece
called
at
the
the bottom
replication
here
fork,
is taking
which
those
is a
little clamps,
Y-shaped
and every
structure
time you
where
start a
new
new
DNA
Okazaki
strands are
fragment,
synthesized
another one of those
by a
clamps is put on the
multi-enzyme
DNA.
complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in
As the
DNA
leading
replication is
strand
the
is
separation of the
moving
two
right into
strands
it.
by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems.
The little sticks
The
sticking
separated
out of the
strands are
lines
called
there
3'
are the bases.
and 5',
So this helps a lot, like to figure out
distinguished by the
how
direction in
Okazaki
which their
fragments
component
go, because you can
nucleotides
see
join
one
up.
being made while simultaneously more lagging
The
strand
3'
template
DNA
is put
strand,
out to
also
get
known
to
as
the
the
other
leading
one.
strand, is
And
diverted
that
to a
answers,
DNA
that answers
polymerase
the earlier
and is
question
used
again
as a
about,
continuous
I
template
was
for the
saying, look,
synthesis
there's
of the
not much
first daughter
room
DNA
for
helix.
single-strand DNA binding proteins on
The
the
other
leading
half of the
strand
DNA
because
double
the
helix,
leading strand
known
is
as the
only
lagging strand,
single-stranded for a
has
very
the
short
opposite
amount of time.
orientation and consequently requires a more complicated copying mechanism.
So molecularly, you
As
can
it emerges
design,
from the helicase,
you can
the
take
lagging strand
this
is
part
organized
right
into
here,
sections
just the
called
enzyme
Okazaki
part,
fragments.
the purple part there, and
These are then
express as
presented to a
E. coli
second
cells,
DNA
and each
polymerase
of
enzyme
those
in the
enzymatic
preferred
heads will
5'
work
to
just
3'
fine by itself.
orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion.
I don't know, maybe I'm too into
The
this, but I
exposed
could just
strands
watch
of
this
single
all day. It's
DNA
so
are
cool.
covered by
No, I
protective
mean,
binding
it's
proteins,
so cool to
and in some
actually see
systems,
that
multiple
what's going
Okazaki
on to
fragments
build
may
all
be
this
present.
stuff are teeny tiny little machines, you know, that are so small that you need to make crystals and do crystal structures to actually see what, you know, to see what is going on. So what the way this worked is that scientists had to get crystal structures of these enzymes in contact with the DNA. So they made structures of them by themselves and in contact with the DNA. Here's the helicase. You can see there's 1, 2, 3 subunits, and the other ones are on the bottom. But this helicase is so processive, it's called, or able to keep going, because it is linked around, you can't see, but it is surrounding not double strand but single strand DNA. So it's right at the point, of course, where the single strand becomes double strand DNA, and it's pushing straight through there. All right. So do you guys want to get together on Monday and have a, you know, and have another little review session at some point? Yes? A what? A Kahoot game. See, if I make it for you and you just play it, right? So why don't you guys make it? If you guys make one of those, and or any kind, like if you make a Kahoot game or a Quizlet cards or whatever, and it gives you links to them once you've made them, then give me the links and I will post them on the Blackboard so that everybody can go use them. All right. And yeah. We are doing it. So there is no class on Monday. During class on Tuesday is the exam. We're having study session right now during class, and then on Monday we'll have to. So what time are you talking? Like what time in the afternoon are you talking about? I could. If I make it at 5, I have to ask my kid to get a ride home from somebody else. So, and you know, it's like she's driving now, and you know, I just made her do that for the bat talk too. So I can make it Monday at 4. Would that be fine? All right. So let's do Monday at 4 in the genetics lab. All right. I'll see you there. And if anybody can't make that, if you can get a couple people together, I'm here all day Monday. Just email me, hey, can we meet and just ask a couple of questions at this time, because I'm back and forth between my office and the lab on Mondays, Wednesdays, and Fridays.